So let's start now today class. So hello everyone, myself Preeti Ranjan, a PMRF researcher scholar from IIT BHU. So today I will take your live session 7 for this problem solving session of this course, computer aided drug design. So let's start. Question number 1. The question number 1 is the molecule A has 14 fingerprints. So molecule A has 14 fingerprints and molecule B has 11 fingerprints. They have 4 fingerprints in common. Calculate the Tanimoto coefficient. So the Tanimoto coefficient equal to C upon A plus B minus C. So in this question, the Tanimoto coefficient is uh, Tanimoto coefficient equal to C upon A plus B minus C. So in this question, A equal to 14, it's given, B equal to 11 and C equal to 4. So we will simply put the value of all this and we will get the answer. So answer will be 0.190. This is the answer of question 1. You all are okay with this question? No reply. So now move to question number 2. Question number 2 is which is not a pharmacophore in this list? So you know what is the pharmacophore? When ligand bind to active site, there is some properties in ligand by which they going and interact to active sites. So the descriptions of that molecular features of ligand is called pharmacophore. So what property we basically see? First one is hydrophobic. Second is aromatic. Third is hydrogen bond donor or hydrogen bond acceptor. And fourth is cations or anions. So these all are the basically 4 features we used to see. So in these questions, they have asked which is not a pharmacophore in this list. So option is given A, aromatic, B, hydrogen bond donor, C, rotatable bond, and D, hydrophobic. So I have written here these all are the features which basically see in pharmacophores. So aromatic, yes, it's match. Hydrogen bond donor, yes. Hydrophobic. So this is the mismatch. This one, so answer will be C, rotatable bond. So now question number 3. In question number 3, if the binding constant for a ligand protein interaction is 2.3, calculate the molar Gibbs free energy at 30 degree in kilojoule per mole. So we know the formula del G naught equal to Gibbs free energy, that is del G naught equal to minus RT ln K. R is here gas constant and which is equal to 8.314 joule per mole. And temperature is given here, how much? 30 degrees Celsius. We should take in Kelvin, so 273 plus 30, this will be equal to 303 Kelvin. Okay, so now we will calculate the del G naught. So del G naught equal to minus 8.314 into 303 into and the binding constant is given here 2.3. So ln 2.3. So 8.314 into 303 into ln 2.3 equal to 0.8329. So this will be equal to 2098.21 and this will come in joule per mole. But they have asked us to calculate in kilojoule per mole. So this will be equal to minus 2.098 kilojoule per mole, or this will be equal to 2.1 minus 2.1 kilojoule per mole. So answer will be minus 2.1 kilojoule per mole. You all are okay with this question? You all are able to listen my voice? Okay, okay. No, because my speaker was very low sound. Okay, okay. So now come to the question number 4. So in question number 4, it's given a relationship is developed between the binding energy of drugs with their activities as PIC50 equal to minus 1.85 minus 0.85 into binding energy. This is the equation, like PIC50 equal to minus 1.85 minus 0.85 into binding energy. So if the binding energy of new molecules is minus 9.0 kilocalorie per mole, calculate its activity IC50 in micromole. Okay. So the binding energy for new molecules is given here minus 9.0 kilocalorie per mole. So we will put the value of this in this formula, like minus 1.85 minus 0.85 into minus 9.0 kilocalorie per mole. So this will come how much? Minus 1.85 plus 7.65 and this will be equal to 5.80 kilocalorie per mole. So now the PIC50 value has come here 5.80 kilocalorie, sorry, not kilocalorie, 5.80. But in these questions, they have asked us to calculate IC50 value and that also in micromole. So the formula here is PIC50 equal to minus log IC50 and that will be equal to log base 10 1 upon IC50. Okay. here it's came PIC50 equal to 5.80. We can write like this also, log 10 1 upon IC50 equal to 5.80, right? Right, log base 10 1 upon IC50 equal to 5.80, or we can write 1 upon IC50 equal to NT log 5.80. So this value will come how much? 630,957.34. So this value is 1 upon IC50. So IC50 will be 1 upon 630,957.34, and this will be equal to 5.18 into 10 to the power minus 6 molar. Okay, and they have asked in question to calculate in micromole. So you all know 1 molar. So this will be equal to 1.8 micromole. You all are okay with this question? Okay. Should I move to next question now? Okay, so this is the question 5. In question 5, sequence A has 400 amino acids. Sequence B has 600 amino acids. Using a blast, a pairwise alignment was done, and 100 and they have find 100 amino acids are identical. So what is the percentage identity? So for calculating the percentage identity, first, we will take this 100 amino acids, and always in denominators, we will take the sequence which has a smaller number of amino acids. So in this both sequence, this sequence A has the smaller number of amino acids. So we will take 400 and then we will multiply by the 100. So this will cancel out and this will be 25%. So percent identity will be 25%. So now come to the question number 6. In question number 6, in problem 5, if 25 amino acids that are different by conservation substitution, what is percentage similarity? A conservation substitution means the chemical properties will be similar. So here they have asked us to calculate the percentage similarity. So what we will do here? Percentage similarity equal to, in previous question it's given like 100 amino acids are identical. So we will take that identical amino acids plus we will add that this 25 amino acid which are different just by the conservation substitutions. So we will add that and then we will take the sequence which has a smaller number of amino acids and then multiply by the 100. So this will cancel out and then 125 upon 4 and this will come 31.5%. So this is the percentage similarity. And now come to the question number 7. In question number 7, it's given that disulfide bonds contribute to protein stability mainly by reducing the conformational entropy of denatured state as del S equal to del S equal to minus 2.1 minus 3 upon 2 into R ln n. So n is the number of residue in the loop formed by crosslink. If the number of residue in the loop increase from 5 to 8, what is the increase in percentage stability? I think somebody again has joined. So in this question, this equation or this formula is given del S equal to minus 2.1 minus 3 upon 2 R ln n, right? And n is the number of residue in the loop. And the number of residue in the loop has increased from 5 to 8, and they have asked us to calculate the increase in percentage stability. R is nothing but this is also a gas constant, and their value is 8.314 joule per mole. So first we will calculate with 5 again. So we will put the value of n. So when n equal to 5, so del S equal to minus 2.1 minus 3.2, 8.314 ln 5. So this will come minus 2.1 minus, we can write 1.5 into 3.14 ln 5 will be equal to 1.60. So this will come minus 2.1 minus 19.9, and this will be equal to minus 22.0. Like this we will calculate with 8. So this is when n equal to 5 and when n equal to 8. So del S equal to minus 2.1 minus 3.2, 3.14 ln 8. What is there? Del S equal to minus 2.1 minus 3 upon 2 R ln n. This is the formula, right? So 3 upon 2, 8.314 into ln 8. So this will be 2.1 minus 1.5 into 3.14 into ln, and ln 8 value will come 2.07, and this will be equal to 27 minus 27.91. So in question they have asked us to calculate increase in percentage stability. So percentage increase in stability equal to percentage increase in stability equal to final value minus initial value upon initial value into 100. So final value has come here 27.91. Initial value is minus 22, minus 22, and again the initial value is minus 22 into 100. So this will come minus 5.91 upon minus 22 into 100. This minus minus will cancel, and then this value will come 591 into 22, and this will come 26.86%. So Answer will be 26.86%. You all are okay with this question? Okay. So should I move next question now? So question number so this is the question number 8. In question number 8, a protein and ligand interact through van der Waals forces. If the ligand moves away by 12%, what is the decrease in van der Waals forces in percentage? So you all are familiar with this equation. Van der Waals forces is related with 1 upon r power 6. So if consider like initial distance is equal to 100, right? So the van der Waals forces will be 1 upon 100 power 6, and this will come 1 e power minus 12. And in questions they have told if the ligand moves away by 12%. So when ligand moves away by 12%, so this distance will become 112, and that time the van der Waals forces will be, and that time the van der Waals forces will be 1 upon 112 power 6. So how much it will come? So this will come 1 upon 1.97382269 e power 12, and this will be equal to 5.06631112 e power 13, e power minus 13. So now we have got van der Waals forces in both cases, like initial cases and after when ligand moves by 12%. So now we have to calculate decrease in van der Waals forces in percentage. What happened? Take, yes? Who have asked something? Someone? Somebody? You calculate this? You just power this, you calculate 112, okay, power 6. You calculate in calculator, you will get, and then you divide 1 upon this, then you will get this value. You understood? uh, this value 112 the power 6, right? So when you will calculate this value, you will get this value 1.9738. I have written a full because don't know, lot of confusion going on, so today I have write it down whole values. So this much came, and then you just divide 1 upon this, so you will get this value. Okay. Okay. So now in this question they have asked us to calculate decrease in van der Waals forces. So decrease in van der Waals forces percentage, decrease in van der Waals forces equal to initial value minus final value upon final value into 100. So initial value has come 1 e power minus 12, and the final value has come 5.06631112 power minus 13. And oh, sorry, not this is the final value. This is initial value only, and this 12 into 100. So the initial value has come this, and the final value has come this. So I have just put it the value of both initial value and final value upon initial value into 100. So when you will calculate, it will come 4.933688 e power 13 upon 1 e minus 12 into 100, and this will come 49.33%. So this is the answer. You all are okay with this question, right? Okay. Should I move? Okay. So now the question number 9. The question number 9 is, what is the uniplot number of the most probable target? The structure is given here, and the hint is given to use Swiss target. So we will go to the Swiss target. This is the Swiss target. So what we will do here, we will draw the structure here, so that we will get the smile. So this is the structure, right? And we got the smile also here, and then simply click in this predict target. Job has submitted here, and we will get the output file after a few seconds. So output file has come here. So the what in questions? In questions they have asked what is the uniprot number of the most probable target, right? So we will see here. This is the target number 1, monoamine oxidase B. This would be the most probable target because it has come in the first number, and the uniprot ID of this target is P27338. So you all are okay? How I have done this? Yes. Okay, okay. Yes, yes. Don't take tension for exam related things that we will discuss later after this class. Okay. Okay. First you focus on class, and after that we will discuss what you will get in exam or not get in exam. Okay. First you focus on this class. So now you are okay with this question number 9, right? So so now this question number 10. When you run a blast search with the above target, what is the question number 10. When you run a blast search with the above target, what is the uniprot number of similar protein which has the highest close identity? So they have asked us to do the blast search with the uniprot number of similar protein, like with the above target. They have asked us to do the blast search with the above target, the target which we have got in question number 9. Okay. And then they have asked us to write down the uniprot number of similar protein which has the highest close identity. So what we will do for this question? Again, we will go there in Swiss target. The uniprot ID which we have got in question 9. This is the uniprot number. on ID. So we will click on this. And after clicking on this, we will directly enter in UniProt, where this protein information is given. In this database, you will get this particular protein information. So all information is given here. You can read from here. But in questions, they have asked us to do the blast. So blast option is given here. So we will click on blast. This is the sequence of this protein. So when you will go down, you will get run blast. So you just click on this run blast. So it start running. It will take few minutes, and then after you will get the result. I have run this file before, so I can show the result. Or should I wait for this to complete? Okay. So this is the result which you will get after completion of that. So this is the ID which we have submitted, right? P27338, right? So the second one, which is like 99.8% similar with this one, is what sequence? H2Q I H6. So answer will be this sequence, this ID. Sorry. So this ID will be the answer. Like in questions, they have asked what is the UniProt number of similar protein which has the highest close identity. So this is the UniProt number of this protein, like H2Q by H6. Now move to the question number 11. In question number 11, perform a pharmacophore model of the following three molecules using pharmagist. What is the number of acceptor in the base code pharmacophore of the pharmacophore the software reports? So in these questions, they have given the three structures, which if you have gone through the SIRS lecture, SIRS has also discussed about this structure in their class. These all are the COX inhibitor, and the name of this structure is validicosive, rafficosive, and siloxosib, like this. Some name is there of these structures. And what you have to do for this, we have to go in pharmagist. Go to this server, right? So in this, they have asked us to upload the input molecules in mol2 format. So what we will do, the structure is given here, we will make it in mol2 format. So for that, what we will do, either we will go to Zinc database, and we can draw this structure. And I have already told the name of these compounds, like name of this compound is valdecosiv, or you can type this name on Zinc database. You will get the structure there, and directly you can download the mol2 file from there. Mol2 file from Zinc database, and make the folder, zip folder, and make it in single zip folder. Like in the one folder, you just make it one zip folder. On that, all three molecules information is available. And then go to this software or this server, pharmagist. You upload file from here and write it down your email ID and submit here. So after few minutes, you and it will take some times, you will get the result. So result will come like this. This is the result. I have submitted this all files and I got the result like this. So in question, In questions, they have asked what is the number of receptor in the best score pharmacophore, the soft pharmacophore. So we can see in the result, this is the score of the all molecules, and in this score, like compared to all, this is the best score, and on this we have to see the acceptor, and we have got only two acceptor. So answer will be 2. I think you all are okay with this question. Yeah, from Zinc database also you can download PubMed, sorry, PubChem. You can download. You can download from anywhere. Yes. Uh, Actually, there is problem. You saw how I have done, right? Result is the problem because you have got, I think, similar questions in your assignment also. Okay. Yes. What is the problem here? Why? Or you can do one thing. If SDF format is there, you can download on that format and go to Open Babel and convert it in Mol2 file. You can do like this now. So you all are okay with this question? So I think now assignment part is over, right? Who has asked? Yes, who has asked question number 1? Why, Derek? Why you came late? So this is the question number 1. Tanimeto coefficient equal to C upon A plus B minus C. You just put it the value of all these. It is all given in questions. A equal to 14, B equal to 11, and C equal to 4. You just put it all value on this equation or this formula. You will get the answer. Okay. Question number. So this is the question number 2. So this all are the pharmacophore properties. So you can see here this 3 has matched with this properties and only this is the mismatch. So answer will be the this. So for these questions also, when you will put it this all values in this equation or in this formula, you will get the answer. Yes. Question number 3 and 4. Okay. So what is the problem on this? Okay. So what happened? You can calculate directly. No, this is the formula, right? Del G minus RT. Like this, you can calculate now upon minus. Huh. PIC 50. Log IC 50. Just now I have calculated na IC 50. So what is the problem? Are, so you can calculate from here na binding energy, right? This is the Gibbs free binding energy, na? Yes. So now you all are okay with this assignment? Okay. 5 and 6. This is the question number 5. In this questions, they have asked us to calculate percentage identity. So for calculating the percentage identity, you have to take first the identical amino acids in nominators and then divide by the sequence which has the lowest amino acids, lower amino acids compared to the other sequence. So in this questions, the sequence A has the lower amino acids as compared to the sequence B. So we will take this 400 and calculate it. This will come 25%. And like that, in question 6 also, they have asked us to calculate percentage similarity. So for that, in previous questions, the identical amino acids has given 100. And in these questions, they have given the 25 amino acids that are differ by the conservation substitutions. So what we will do here, we will just add this 25 amino acids also in that similar amino acids. And we will divide by the 400, the sequence which has the lower amino acids into 100, and you will get the answer. So I think you all are okay with this assignment, right? Which question? Huh? 11. Yes, tell me what is the problem in this question. What is how the? So for these questions, just now I told you have to take this as you have to draw this as structure in zinc database. If you don't know the name, I already given the name of this all three structures is here. So you can simply type this name in Zinc database. You will get the structure, and from there you download. Either you download in Mol2 format or SDF format. So if you will download in SDF format, you just convert it in Mol2 format because this particular software, like the Pharmagist, they require the file in Mol2 format. So you have to convert these things in Mol2 formats by using OpenBabel or any other softwares. After that, you make one zip folder by keeping this all three molecules in one folder, and you just put it in this folder. Like they have asked us to choose files, so you just choose the files and give your email address and submit it. After few minutes you will get the result in your email. You will get the result like this in your email. Okay, and from there whatever ask in your question you can write it down. Yes, it is not visual here. Are you can go search lecture now, you will get the structure name. Why you people are not reading at all or what? Okay, so this is the name of this all three molecules. So now you all are okay with these questions. Okay, 1 minute. Oh yeah, yeah. You just wait. Yes, yes. You just wait. Yes, yes. Karthik, you have joined here. Okay, so a student. Okay, okay. 1 minute, Karthik. 1 minute. So a student. This is Karthik, and he will tell about your whatever difficulty related to your exam and all things. So he will explain to you all these things. Okay. So whatever doubts you have regarding exams and anything, you ask now. Karthik, you check chat box also. I think some students are asking in chat box. but the, Sri Devi, but this all video is available in NPTEL site now. Yes, in problem solving session you go and you will get from there. Yes, yes. For the live sessions, what I have told for these questions, there is one mistake. So what I have shown in this discussion forum, like 23.98, that is the correct way. You have to do like this. No, these all are the correct. See, the only problem in question number two, and I think the question number three is the similar like question number two in assignment five, right? Yes. So in assignment five, the only question number two, there is a problem, and you people also didn't see. I didn't see there, so I made one mistake. So in place of 6.01, you have to take 5.99 and 6 and calculate it. If you have there in class, and you have seen like in question number 4, 5, 6, how I have solved. Like I have taken the small step length and I've calculated and reached the final steps. Like, so if you are in question 4, 5, 6, and there the 6 to 6.1, right? In question number 6, they have decreased the step length and we have taken the smaller step length and we have reached finally the 6.1. But in question number 2, they have asked us to calculate at 6. So the final destination was there 6, and we have taken 6.1. So this is the mistake. So what has given there in discussion forum the solution for this question number 2, that is the correct, and you just take that one and go through that. And other are correct, I think. Or if you people want, I can show in next class that question, correct solution. Okay. Okay, fine. I will show in next class. I thought that is enough because already shown there in discussion forum, no? Yeah. So yeah, that's why this class I didn't bother about that because you already posted there in discussion forum the correct method. And no, Karthik, and one more thing is there. Not only that question, the question number 4, 5th, and 6th, all are similar questions, and all there I have taken only that a step length question is there. So if they will, so once they will learn the concept, if what concept is going on, they can rectify themselves. Nothing new in question 2. Yeah, but the concept is similar. Na? They have taken a step length only. I'm I am focused on a step length. Yeah, method is there. They have used different formula there, but the step length things because the no the only problem is Karthik. The question number 3, they have asked us to calculate up at x equal to 6 in question number 2, and they have given the step length 0.1, right? In question number 3, they have asked us to just change the a step length and ask us to calculate at 6. So question number 2 and 3 is similar questions, only the step length was different. But in question 4, 5, and 6, I think they have asked us to calculate at 6.1, and the step length was given 0.1 in one questions, and in other questions given 0.025 and something like that. So my question is here, like they have just changed the method. In question number 2 and 3, they have asked us to calculate by 2 point definite method something, and question number 5 and 6, they have asked by some other method. But the concept is similar. So if the people will learn the concept how to do that, they can rectify it themselves. Yeah. Y- So now your students, your all examination related issues solved. No, no, I don't have. I don't think. Yeah, I think it's given in NPTEL site about exams and all, what marks and all. You can see from there, no? From NPTEL sites. Oh, I was not aware of this, Karthik. That's why I told them to use another. I think all students' problems has solved. So we should close the class. Okay. Okay. Thank you, Karthik. Okay. Thank you.